Welcome to Lecture 9, in which we will go over the process of meiosis. Our learning objectives for this video are to distinguish between somatic cells and gametes, describe the overall process of meiosis 1 and 2, and know their end result, describe the process of synapsis and crossing over during prophase 1 of meiosis, understand independent assortment, and define non-disjunction and understand its effects on ploidy. By the end of this video, you should have an idea of the similarities and differences between binary fission, mitosis, and meiosis. In the previous lecture, we talked about mitosis, which involves the division of somatic cells. Meiosis, on the other hand, is cell division that produces gametes. Somatic cells are non-reproductive cells, which would be most of the cells in our body. In humans, somatic cells are diploid. Gametes are reproductive cells, like the sperm and the egg. Unlike somatic cells, they are haploid, so they only have one copy of each chromosome. Gametes are derived from germline cells, which are diploid cells that divide and form haploid gametes. In sexual reproduction, two haploid gametes, specifically a sperm and an egg, combine in a process called fertilization. This produces a zygote, which is now diploid because it received one copy of each chromosome from the sperm and the egg. Over time, the zygote will undergo mitosis and produce more cells, and the germline cells in the testes and the ovaries will undergo meiosis to produce more gametes. This continues the sexual life cycle. Meiosis is the process that germline cells undergo to divide and produce haploid gametes. It is divided into two phases, meiosis 1 and meiosis 2. Like mitosis, it begins with interphase and has the same phases of G1, S, and G2, in which it grows and replicates DNA. This diagram shows two pairs of homologous chromosomes, with the gray chromosomes coming from one parent and the red chromosomes coming from the other parent. Once the DNA is replicated, we still have two pairs of homologous chromosomes, but each one is now duplicated. Once the DNA is replicated, we still have two pairs of homologous chromosomes, but each one is now duplicated. The remaining phases of meiosis are also similar to mitosis, and whether it is part of meiosis 1 or 2 is just specified by adding a 1 or 2 behind the phase. For example, the equivalent of prophase in meiosis 1 would be prophase 1. Meiosis 1 is the first round of meiotic division. This is when the homologous chromosomes are separated from one another, creating two haploid cells with one copy of each duplicated chromosome per cell. Notice how these chromosomes do not look the same as they did prior to dividing. This is because of some special events that occur specifically during meiosis 1. The first is that during prophase 1, a process called synapsis occurs. This is a process in which each pair of homologous chromosomes are brought together and physically joined into a synaptonemal complex, also referred to as a tetrad due to the presence of four sister chromatids, or a bivalent. Similar to the previous image, the different colors represent which parent each homologous chromosome is from. As usual, the sister chromatids are connected by the centromere and have kinetochores that will be used to separate the copies later on. The two chromosomes are connected by filaments. Another event that occurs during prophase 1 is crossing over. This is the process of homologous chromosomes crossing over as seen here and exchanging the genes in these segments. In the image, the letters represent different genes. Both chromosomes have genes A, B, and C, but the genes from each parent are slightly different, and this is represented by one chromosome having uppercase letters and the other having lowercase letters. During crossing over, the exchange of genetic material creates a chromosome that now has genes mostly from one parent, but some of the genes now come from the other parent, and vice versa. This process is also called genetic recombination, as the genes are essentially being taken off from each chromosome and being recombined into a different pattern. The chromosomes that crossed over and have a mix of maternal and paternal genes are referred to as recombinant chromosomes, while the chromosomes that did not cross over are the non-recombinant chromosomes. This process helps increase genetic diversity. The point at which the chromosomes cross over is called the chiasma, or chiasmata for multiple points of contact. The chromosomes remain attached at the chiasmata until they separate in anaphase 1. The remaining steps of meiosis 1 are similar to mitosis, with a few exceptions. Like in mitosis, the nuclear envelope begins breaking down in prophase 1, and the mitotic spindles attach in prometaphase 1. However, at metaphase 1, the homologous chromosomes are lined up as pairs, instead of in a single file line of chromosomes. Then at anaphase 1, the homologous chromosomes are the ones that are separated from one another, not the sister chromatids. This results in two haploid cells that have just one copy of each chromosome. The two sister chromatids that make up each chromosome are no longer identical due to crossing over. Telophase 1, like telophase in mitosis, involves the nuclear envelope reforming and the mitotic spindle breaking down. Meiosis 2 is the second round of meiotic division. This is when the sister chromatids separate, resulting in 4 haploid cells. There is no DNA duplication event between meiosis 1 and meiosis 2, allowing the cells to remain haploid. Meiosis 2 is very similar to mitosis, with the only difference being that the cells are haploid. The nuclear envelope breaks down, and mitotic spindles form again in prophase 2. The mitotic spindles attach to the kinetic cores of the sister chromatids in prometaphase 2. The chromosomes line up in the center in metaphase 2. The sister chromatids separate in anaphase 2, and the nuclear envelope reforms while the mitotic spindle breaks down in telophase 2. After telophase, cytokinesis breaks the cells apart. Since there were 2 haploid cells from meiosis 1, meiosis 2 ends with 4 haploid cells. Independent assortment refers to the idea that when chromosomes and sister chromatids separate during meiosis 1 and 2, how each pair is separated and assorted into the dividing cells is independent of how the other pairs are separated. Whether a cell receives the maternal or paternal copy of a chromosome in meiosis 1, or the recombinant or non-recombinant copy of a chromosome in meiosis 2, is completely random. This diagram shows an example of how this would look in a cell with 2 pairs of chromosomes. Let's call the smaller chromosome on top chromosome 1, and the slightly larger one chromosome 2. When the homologous chromosomes divide, there are 2 possibilities. In the first possibility, the paternal copies of chromosome 1 and 2 end up in one cell, and the maternal copies end up in the other. When division happens again during meiosis, whether a cell gets only recombinant copies or non-recombinant copies, like in outcome number 1, is equally likely as a cell getting a mix of both. Likewise, it's also possible for there to be a mix of the maternal and paternal copies after meiosis 1, and again, the separation of the recombinant and non-recombinant chromosomes is random. For a diploid organism with 2 chromosomes, like in this diagram, there are 4 total possibilities. Humans have 23 unique chromosomes, so there are 2 to the power of 23 possible combinations, which comes out to over 8 million possibilities. Like crossing over, independent assortment contributes to the genetic diversity that humans have. Sometimes errors occur in the separation of homologous chromosomes or sister chromatids during anaphase 1 and 2. This results in non-disjunction. If non-disjunction happens during meiosis 1 and a pair of homologous chromosomes fails to separate, this results in one cell having an extra copy of a chromosome and another cell missing a copy of a chromosome at the end of meiosis 1. When the sister chromatids divide, this results in 2 cells with an extra copy and 2 cells missing a copy. If the homologous chromosomes separate properly and non-disjunction instead happens in one of the 2 cells during meiosis 2, it results in one cell having an extra copy and another cell missing a copy, but 2 cells that still have the correct number of chromosomes. Non-disjunction leads to aneuploidy. Remember that ploidy refers to the number of chromosomes an organism has. An individual that is aneuploid has an incorrect number of chromosomes. Monosomy, with mono meaning 1, means that a chromosome is missing, leading to there only being one copy of one of the chromosomes instead of 2. This ends up being fatal, and zygotes with monosomes are unable to make it to birth. Trisomy, with tri meaning 3, means that there are 3 copies of one of the chromosomes instead of 2 copies. Like monosomies, some are fatal, but some trisomies are able to make it to birth and live a relatively normal lifespan. Trisomy 21, also known as Down syndrome, is the most common trisomy. This is a karyotype of a male with trisomy 21. There are 2 copies of all of the chromosomes, except for chromosome 21, which has 3 copies. In this video, we learned that somatic cells are diploid and non-reproductive cells, while gametes, like sperm and eggs, are haploid reproductive cells. A sperm and an egg will combine during the process of fertilization to form a diploid zygote. Meiosis is divided into 2 phases, with 2 haploid cells produced at the end of meiosis I and 4 haploid cells produced at the end of meiosis II. Prophase I has 2 unique events. One is synapsis, which is the physical attachment of the homologous chromosomes together. The second is crossing over, which is when the homologous chromosomes exchange genetic material, increasing genetic diversity. The homologous chromosomes and sister chromatids separate independently from one another during anaphase I and II respectively. This creates many gene combinations that further increase genetic diversity. Non-disjunction is an error that occurs when the homologous chromosomes, or sister chromatids, fail to separate properly, and this leads to extra or missing copies of chromosomes. In the next lecture, we will begin discussing genetics and how genes are inherited. Thanks for watching.